which you also know as the cell membrane. So back when we were in grade 10 and we talked about the cell, we would draw pictures of the cell or label pictures of the cell, and we would point to this structure here and say, yes, cell membrane. And all of my students in grade 10 would nod and say, for sure, that's the cell membrane. And you might remember from grade 10 that the function of that cell membrane, we said, was a barrier. It separated from the inside of the cell from the outside of the cell. And all the grade 10 said, yes, of course, that makes sense. And we said it also lets materials be transported into and out of the cell. And everyone nodded again and said, yes, that makes sense. But what all my grade 10 students always forget to ask is, well, what is the cell membrane made up of? Is it really just this line around the cell? And they forget to ask how. How does that membrane control substances going into and out of the cell? So now that you're in grade 12 and you have these advanced critical thinking skills, I know when we were pointing to that cell membrane in the cell, you were thinking to yourself, well, what is it really made up of? How does it work to transport things into and out of the cell? I know you've gotten to that point where you're now asking that. And so this is kind of where our whole first unit ties together. So why did we learn about all those macromolecules? Why did we learn about proteins? And why did we learn about carbohydrates? And why did we learn about lipids? And how is this going to really apply to biology? Well, here we go. So grade 12 functions of the cell membrane are still very similar to what we said in grade 10. The cell membrane is a physical barrier. It separates the inside of the cell, which we would call the cytoplasm, from the extracellular environment. So the fluid that surrounds cells we call the extracellular environment, or the extracellular fluid, or the extracellular matrix. It comes kind of with a lot of terminology. And this barrier separates the inside of the cell from the outside. But what we want to focus on for this course is this regulation part. The cell membrane is going to regulate the movement of substances into and out of the cell. And this is going to link to what we're going to talk about next unit. We're going to talk about things like cell respiration and photosynthesis. And to do those processes, cells need to move substances in and out. And so how does it do that? And that's going to be the focus of what we're talking about in this part of the unit. So what is the cell membrane? We kind of alluded to this a little bit when we were talking about the macromolecules. We talked to you about how phospholipid bilayers form in solution. And so what we have up here in the corner is this bilayer structure. You've got amphipathic phospholipid molecules. And remember that word, amphipathic. It means that those polar heads that are facing the outside and facing the inside are hydrophilic. They're polar, they like water, and they're going to be facing the aqueous or watery solution inside the cell and outside the cell. In contrast to those polar hydrophilic regions, you've got the inside of the phospholipids, those fatty acid tails, that are hydrophobic and nonpolar. They do not want to mix with water. They don't do well with water. And so they're going to hide themselves inside this bilayer structure away from the watery areas outside the cell and inside the cell. And so when this happens, and this kind of spontaneously forms in solution, the phospholipids will arrange themselves in this structure. You get the beginnings of the cell membrane. And so how do we know about this structure? How do we know what it looks like? Well, there's some scientists from the US, Singer and Nicholson are their names, and they proposed this model in 1972 and gave it the name the fluid mosaic model. So remember that term, fluid mosaic model. You're going to hear that over and over. It's a very famous model. You'll see it over and over again in your biology studies. And according to this model, I want you to pay attention to the words fluid and mosaic. Those have meaning. So according to their model, the membrane is a mosaic. It's going to be made up of these phospholipids that we talked about earlier, but it's also going to have proteins embedded. It's going to have carbohydrates. It's going to have cholesterol. So this mosaic part is this coming together of different components to make this membrane structure. And the fluid part, I want you to really remember that this membrane is dynamic. It's in motion. It's moving all the time. So it's a fluid layer of phospholipids that these other components are going to be embedded. So let's take a look. What does the cell membrane look like? Here it is. Here's the fluid mosaic model. So when you were shy in grade 10 and didn't want to ask that question, what is it really made of? Here it is. So this is what we think. And all cells kind of have this structure, this fluid mosaic model. To different degrees, they have different components arranged in different ways, but the main components that are there are all the same. So let's take a look at, first of all, what are we looking at? This is the phospholipid bilayer. So you see the polar heads of the phospholipids facing outside the cell. You see the polar heads of the phospholipids facing the cytoplasm of the cell. And you see the fatty acid tails kind of hiding out in the middle, in that sandwich in the middle of the layer. So this is bilayer. You've got this top layer and you got this bottom layer. So 2 layers is the bilayer structure. And it's going to form spontaneously in aqueous solutions, getting those fatty acid hydrophobic tails away from the watery portions, orienting the polar heads towards them. Membrane is around 8 to 10 nanometers thick, so it's not a thick structure. But again, and as we're going through this, remember structure, function. How does the structure reflect the function? So we've got a lot going on in this diagram. You've got that kind of basic phospholipid bilayer. You've got these components stuck through the bilayer. You've got this one winding through the bilayer. You've got what look like carbohydrates attached at the surface. You've got a lot going on here. What are these molecules embedded inside the bilayer? So let's take a look. Let's take a look at all of these components. Okay, so we talked about this. Hydrophilic phosphate heads, hydrophobic fatty acid tails forming that structure. And now if we look at the components themselves, you'll see that these big transmembrane structures that are kind of spanning the whole bilayer, these are proteins. So the orange components are proteins. Some of them go all the way through. And if we think about this for a minute, the center of the bilayer is hydrophobic. So if these orange structures are coming all the way through the bilayer, then the amino acids in those proteins in this particular section must be hydrophobic. They're going to mix well with this nonpolar interior. And then you're going to have amino acids at either end that are going to be hydrophilic, that are going to mix well with water. So the structure of this protein, how the amino acids are arranged, is going to make it fit as a transmembrane, or we also call it an integral protein. And that's going to come right through the bilayer structure. And it can do that because of the hydrophobic amino acids inside, the hydrophilic amino acids towards the outside. Okay, so we've got these that span, and I want you to kind of think about, well, what would be an advantage of that? What would be a function that these proteins could serve if they span the entire phospholipid membrane? You're also going to see these green structures. They kind of look like carbohydrate structures, and they are. So these are carbohydrates, and you've got some of them that are attached directly to phospholipids. So you've got this right by the polar head of the phospholipid. And so if a carbohydrate chain is attached to a phospholipid molecule, we call it a glycolipid. Okay, so remember, glyco always refers to sugar or carbohydrate. So if I've got this structure attached to a lipid, it's a glycolipid.
Okay.
Sorry. And then, okay, over here you can see you've got the carbohydrate chains attached to a protein, attached to the orange molecule. And so again, it's a glycoprotein. It means a protein attached to a carbohydrate structure. And again, the glyco means carbohydrate. Okay, if you look inside the membrane, you'll see this structure. And if you remember the 4-ring structure of steroids, that's a cholesterol. And you'll see that those are embedded through the phospholipid bilayer at various locations. And so that's going to be important in terms of the fluidity of the membrane. So again, take a look at the structure. You're going to be expected to be able to label it. You're going to be expected to be able to draw it and label it on your own. This is a critical diagram. So know the phospholipid bilayer structure. Remember those short form for phospholipids, the circle for the polar head, squiggly tails for the fatty acids. That's forming kind of the basis of the membrane. Embedded in that, you've got these proteins. Some of them travel all the way through. Some of them are only at the surface. So we've got a couple of terminology, bits of terminology for that. If it goes all the way through, it's called an integral protein or a transmembrane protein. If it's only associated with the surface, the inside surface or the outside surface, it's called a peripheral protein. So I know they don't label that on this diagram, but these ones that don't span would be called peripheral. Okay, so there's a fluid part of the fluid mosaic model. And so you should know, if we go back to this diagram, these phospholipids can flip back and forth. So they can laterally move very quickly, many times a second. And so they're in motion. So this is constantly moving. I know it's hard to tell from a diagram. It looks pretty static here, but it's actually very dynamic. It's moving. Those phospholipid heads, the polar heads, are kind of rotating back and forth. Less common is actual flip-flop motions. Okay, so it's not very often that you'll see a phospholipid come all the way through the membrane to the other side of the bilayer. That's hard to do because those polar heads have to go right through the hydrophobic middle, and that's not easy chemically. So you'll see that rarely. Okay, sometimes it happens, but not as often as this lateral movement that you get all the time. Temperature is also going to influence fluidity. So we know about chemistry, about kinetic energy. The higher the temperature, the greater the motion in those molecules. So you increase the temperature, the molecules are going to move around more. So that's going to increase the fluidity of this membrane as well. The other thing that increases fluidity, and let's talk about structure again, we talked about saturated versus unsaturated fatty acids. Remember the hydrocarbon tails? If they're saturated, they're going to stack. And if they're going to stack, they're going to be kind of solidifying. If they're unsaturated and you've got that double bond that makes the kink, then those fatty acids aren't going to be able to stack on top of each other anymore, and you're going to have more fluidity inside that bilayer. So the more unsaturated fatty acids you have, the more fluid your bilayer is going to be, the more dynamic, the more in motion it's going to be. And so we want that. We want this fluid model for our cell membranes because the more in motion it is, the easier it is to transport things across. So the other thing, we talked about the cholesterol molecules that were inserted into the bilayer. Those also influence fluidity, and they have different effects depending on the temperature. So if cholesterol is inserted and it's high temperature, it's going to calm the bilayer down, reduce the phospholipid movement. If it's colder temperature, we're going to worry about these molecules slowing down and solidifying. Cholesterol inserted in there prevents that. It's going to disrupt the packing. Those phospholipids are going to stay mobile, stay fluid. So cholesterol is really important in maintaining fluidity as the temperature changes around the cell. Proteins are also moving. It looks weird that they're those big structures stuck into the membrane, but they can also move. They can drift through the membrane. And so when you're thinking about this membrane, think about it in motion. Take a look at some of the videos that I posted on Classroom that shows this kind of dynamic nature of the cell. The cell's not just sitting there. Things are moving. Things are being transported. So all of these things can move through and contribute to the fluidity of the membrane. The mosaic part of the name, and if you take art and you know about mosaic art, it means you're putting different components together. So the cell membrane's a mosaic because we've got all these different components. We've got phospholipids, we've got proteins, we've got cholesterol, we've got carbohydrates, all working together to form that barrier, that structure that's going to allow things to be transported into and out of the cell. So here's where we're applying these structures that we've learned about in the beginning of the biochemistry unit. All of these are incorporated into the membrane structure. So all cell membranes differ in the nature of their mosaic, where the proteins are, how many proteins, what kind of integral proteins, what kind of carbohydrates are on the surface, and that's reflecting the different functions of each of the cells that they're part of. So again, structure reflecting function. So you may have also remembered the term that the cell membrane is selectively permeable. Sometimes it's called semi-permeable, means the same thing. And it's kind of, if you think about the word selectively, it means it's going to allow some substances to cross more easily than others. So the membrane is going to be selective about what it lets go in and out of the cell. So if you're a hydrophobic substance, and remember we talked about like dissolves like, so if you're hydrophobic, you're going to pass readily through that bilayer. They're soluble in the lipids. Those fatty acid tails are going to be no problem. Hydrophobic substances generally can pass through that phospholipid bilayer. But you have a problem if you get hydrophilic substances and charged particles like ions. So how is a hydrophilic substance, a water-loving substance, a polar substance, going to get through that fatty acid interior of the bilayer? That's going to be really hard. Like dissolves like. Hydrophilic doesn't work with the hydrophobic phospholipids in the center. So that's where those proteins come in. Those proteins, those integral proteins that span the whole membrane, are there to help transport things that are hydrophilic and things that are charged, giving them that pathway through the membrane into the cell or out of the cell. So that structure is there. It's important for the function of the cell. It's going to need hydrophilic things. Hydrophilic things can't pass through that phospholipid bilayer because they don't mix with the fatty acids in the middle. So the proteins come in and say, here, let me provide a pathway. So if we take a look back, you can imagine, You can imagine hydrophobic things just floating right through, diffusing through generally. We'll talk about different mechanisms of transport next, but they're going to come through the phospholipid bilayer. They're hydrophobic. They mix well with the fatty acid interior. They're going to come through fine. Hydrophilic stuff is going to look for a protein. They can't get through this hydrophobic interior. They're going to find a protein that gives them this passageway through. And some of these proteins act like tunnels that let them come through. Some of them act like transporters that move them through. And we'll talk about how those proteins function to move things across as we go on in this section. Okay? And so what you're going to look at is the function of all of these different things. Why are there carbohydrates on top of these proteins and lipids? And it turns out these carbohydrates are kind of cell recognition signals. They're signals for things that are outside the cell to kind of identify what this cell is. These are some of the markers that you'd have on your red blood cells to tell you what blood type you are. So these carbohydrates tend to be cell signaling and cell-to-cell communication markers. The proteins, transporters, some of them that are just at the surface, the peripheral proteins, those are enzymes. Sometimes they carry out enzymes, reactions that are happening at the cell surface. Those are enzymes to help catalyze them. The cholesterol for fluidity, the hydrophobic fatty acid tails to form that bilayer structure, the polar heads to face the watery surfaces outside and inside the cell. And this underneath here, the cytoskeleton, these are more structures that are giving anchoring kind of architectural support for the structure of the cell and for the membrane. So those structures are there as well. Okay, so part of what you're going to do as you go work on your own through this section is kind of identify all these structures and then relate what the function is for each of those. And I hope that helps you understand the cell membrane.